Hi guys, we're gonna sassy. We're gonna be working on chapter 8 in microbiology. Our chapter today is bacterial genetics. Okay, so I've taken lots of notes in my handy dandy notebook. I've filled up half the notebook with the chapter. So I'm hoping, sassy, hush. So I'm hoping we're gonna get through this quickly. Shh, shh, hush. That's my security system. All right, so the first person that we're going to talk about is McClintock. And this person studied corn kernels, and they studied the color and learned about something called transposons, okay? I'm going to mess up the name of that word, so just forgive me. So transposons are also called jumping genes, and DNA was moving into and out of the genes. So essentially, it was transposons was jumping in and changing the corn kernel to, I don't know, yellow, and then jumping to something to another part was moving into and out of genes. So, and moving into another gene and then causing the corn to be, I don't know, white, okay? All right, next. Well, we're going to talk a little bit more of that. We're going to build into it. But that was the glimpse of history. Okay, so McClintock studied corn kernels and learned about transposons. Moving on. Natural selection. So it's adapt or die, right? We've heard about this a lot. Otherwise, adapt or die. Otherwise, others will take over. Okay, so if all these bacteria die, other bacteria will come and fill that gap, okay? Two ways bacteria can adjust to survive, okay? So two ways that these bacteria can survive. One is through regulating gene expression, and the two is genetic change. And we're going to break those down. E. coli used more than... Sassy, hush. E. coli used more than any other organism in the world to look at, okay? Why? Why do they use E. coli? It's because they grow fast, it's easy to grow, and they grow in large quantities, large numbers. And so now you can study rare events and why strains differ in genetic makeup. All right, next. A change in genotype and a change in phenotype. Genotype is the genes. Phenotype is physical traits. It's what you can physically see. Okay? So, temperature, sassy hush. Temperature can change a phenotype. I found this to be fascinating, that a temperature can change the phenotype, but not the genotype. So just because it changes the phenotype, it doesn't necessarily mean that it changes the genotype, okay? So make sure you know that, because that can be something that kind of hangs you up. Temperature can change the phenotype. So for instance, kind of think about a lizard. You know how a lizard changes colors depending on the temperature? So it's changing its phenotype, but it's not changing the genes. It's not changing the genotype. A change in organisms' DNA... A change in an organism's DNA alters its genotype, the sequence of nucleotides in the DNA. So that's the bold-faced word, genotype, which is a change in an organism's DNA alters its genotype, the sequence of nucleotides in the DNA. So if you're changing the sequence of nucleotides in the DNA, you can change the genotype. Phenotype is observable traits. We've talked about that, like what you can see physically, phenotype, physically see. Okay, genetic change. Genetic change can happen. We know that. How can it happen? Two ways. One, mutation, and two, horizontal gene transfer. Mutation, change in nucleotide sequence. Okay, so think about this. Remember we talked about the A's and the T's and the C's and the G's in chapter 7? So the way that I can remember is those A's and T's are the A is the base, Remember, you have the nucleotide, you have the base. The base has the A in it, and then you have a sugar and phosphate. So a mutation is the change in the nucleotide sequence. So an example would be, instead of an A and a T, it might have changed to an A and a C, okay? That would be an example of a mutation where the nucleotide sequence got changed. And so then it gets passed to a progeny. What is a progeny? A progeny is a daughter cell. So pretend I'm a parent. I can pass that sequence, that mutation, on to my children, okay? And you do that through vertical gene transfer. So now you can have what I call a little mutant baby, okay? So let's talk about that again, because we're going to be talking about mutations all the time. So a mutation is a change in nucleotide sequence. An example would be AT to maybe AC. Then it gets passed to a progeny, which is a daughter child, through vertical gene transfer, and now we have a mutant baby. Okay, next, horizontal gene transfer. What the heck is that? Horizontal gene transfer is movement of DNA to one organism to another. So pretend like this is a gene, this is a cell, and this is a cell, or this is a bacteria, this is a bacteria. They come together, and genes gets transferred to from one to another horizontally. Okay, it's kind of self-explanatory. We can then mutate by giving birth to a prodigy, okay, by vertical gene transfer, creating a mutant baby. From that, all started by horizontal gene transfer. So remember, there are two ways that we can do genetic change. One is mutation. Two is horizontal gene transfer. Mutation is we're changing the nucleotide sequence and we're passing it to our prodigy, our little baby mutant, and we're doing that through vertical gene transfer. Horizontal gene transfer is going from one to the other, again passing it to our prodigy, our little mutant baby, and that's happening through vertical gene transfer, okay? Mutations and horizontal gene transfer are genetic changes that both use vertical gene transfer. Got it. Okay. Moving on. Auxotroph. What the heck is an auxotroph? An auxotroph is a mutant that requires nutrients. Okay? It requires nutrients. What could that nutrient be? I don't know. That nutrient could be magnesium, or that nutrient could be, I don't know, specific protein. We'll go through it a little bit more. But it requires a nutrient, okay? Photoautotroph is a mutant that does not require a growth factor. A photoautotroph is a mutant that does not require a growth factor. An example of an autotroph, a gene is required to make tryptophan. Okay, so let's say you have a gene, you have a cell, and a bacteria. Let's talk about bacteria. We have a bacteria. That bacteria's name is autotroph, and it requires tryptophan. Tryptophan is found in chicken. Okay, I believe it's found in chicken. And let's say it's deleted. Oh no, you know, the gene that that was making tryptophan so that cell could stay alive, now it's gone. So now, as a result, the organism can only survive if it's supplied with tryptophan. So think about it. Let's say you have some gut bacteria in your stomach, and that bacteria was making its own tryptophan, and it had the genes, and it was rocking, and all of a sudden, bloop, it got deleted and it can't. So the only way for it to survive is if I eat some chicken and then it gets bathed in tryptophan and it can eat, okay? Next. Moving on, wild type. This is very important. Wild type is a particular phenotype isolated from nature. An example would be wild E. coli. Wild E. coli is a photoautotroph. Can that be right? That's what I put down. I hope that's right. I thought that this was kind of confusing. A wild type is essentially what would happen naturally in the wild, okay? So if, for instance, you can have chicken pox, okay? So you can have wild chicken pox, or you can have a vaccine that has chicken pox that's not wild, okay? They manipulate it, okay? So wild type is what, you know, if it's a bacteria, if it's a virus, whatever it is, it happens naturally, okay? So it's whatever happens naturally in there. For this example, they were talking about wild E. coli. All right, next. Strain characteristics are designed by 3-letter abbreviations. First letter capitalized. A strain that cannot make tryptophan. So I think I should just show you right here. Let me find it. Okay. So do you see right here the first letter is capitalized, and then it's 3 letters, and so it's T, R, P, and then that little negative. So that little negative means that bacteria, so it could be TRP negative, and then it could be E. coli, for instance. And so that means that only required growth factors are indicated. So that bacteria would need tryptophan in order to survive. If it didn't, I think it has a plus, but we'll get to that. So it's capital TRP. When it has the negative, it's only required growth factors are indicated. So those are the tryptophan is what that bacteria needs in order to grow. So if it's resistant to an antibiotic, it's going to show, let me show, the capital STR, and then it has a little R showing that it's resistant. You can also have an S, and I believe that means it's sensitive, but I couldn't find it in the book, so you might want to double check that, but I think I'm right. Okay. Spontaneous mutations, genetic changes that can result from normal cell process. We know that. Cells multiply, and as they divide, you can have a spontaneous mutation. It happens naturally. Sometimes, but it's infrequent. It doesn't happen that much. So it's like 10 to the negative 4th, 10 to the negative 12th. If you want to add it up, you can add it up and do the math right here, but it's pretty basic, okay? So yeah, like you just add 10 to the negative 4th plus 10 to the negative 12th, and that would be 10 to the negative 16th, okay? No big deal. I think I do an actual math problem. Yeah, so we did that on the next page. Okay, reversion. What is reversion? Reversion, a mutated cell goes back to non-mutated state. The heck you say? So it gets mutated and it can go back to its state, and that's called reversion? Whoa. This acquires spontaneously at low frequencies. So just as it can mutate, and that's rare, it can revert, something called reversion, and that happens very little. Next, mutations can happen routinely. So although it doesn't happen a lot, it does happen routinely. In a large population, you are bound to find a mutant. So technically, in a colony, they aren't all identical, technically. But remember, the true definition of like colonies, it comes from one. But technically, they're not all identical. if you have a mutated cell and the environment changed, then that mutated cell might be better suited for the new environment. Okay? So let's say, for instance, there was a, let's say for instance, there was, everything was green grass, everything was green, and you had this bug, and the bug was green. And then it mutated into white. Well, that's a disadvantage, but the environment changed and everything is all white, so it would be an advantage. Okay? So sometimes mutations can benefit, is really the bottom line. A mutated cell might be able to be antibiotic resistant. What happened was the antibiotic killed all the sensitive cells, allowing resistant cells to grow without competition. So remember how we said something else will take into place? So you have all these bacteria that are multiplying, right? And you have these little mutants, and they have their little mutant babies, right? And how are they doing that? Through vertical gene transfer. And so you have a couple of these little mutants and these little mutant babies, and then all of a sudden you have an antibiotic and it goes bam. And so there was all these bacteria, but now they die. So now there's only a couple little mutants and, you know, one or two little mutant babies, but now they have the opportunity, because all those other things are dead, to grow and grow and grow and grow. And so that's how antibiotic resistance happens. It's not creating the mutations, because the mutations just happen naturally. What's happening is it's killing all the sensitive cells and letting the mutants kind of take over, okay? So for an example, if you give, oh, well, here's something different. All right, let's move on. Give two drugs. So let's say you get, I don't know, bacteria infection. For some instances, you can give two drugs. And when you give two, you do this because it's unlikely that the cell will mutate and be resistant to both medications. So if you have this bacteria, and let's say, I don't know, the antibiotic, the first antibiotic does something to the cell wall, and the second antibiotic does something inside, the likelihood that that bacteria can mutate both for something inside and both something outside, for example, it's not likely. It does happen, but it's not likely. That's why they double down on the antibiotics. Okay. So the mutation rate to an antibiotic, for instance, is 10 to the negative 6 per cell division. And you have another antibiotic, and that's 10 to the negative 8. Then the probability of both mutations happening within the same cell is 10 to the negative 6. 5, 10, it should be plus 10 to the negative 6 plus 10 to the negative 4, which is 10 to the negative 14. I wonder if that's how you would, being that you're adding it. So if its resistant rate is 10 to the negative 4 and the other is 10 to the negative 8, then the probability of both mutations happening at the same time would be, you know, essentially you're just adding them. It's simple math. Okay. Next, we're going to move on to something called base substitutions. Base substitutions are the most common type of mutation occurring during DNA synthesis when an incorrect nucleotide is incorporated. What? Base substitutions. So let's remember what base substitutions are. Base substitutions are like A, T, C, G, right? So let's say you take out an A and you put in a C. That's a base substitution. And the most common type of mutation occurring during DNA synthesis when incorrect nucleotide is incorporated. So remember the nucleotide, you have the sugar, the phosphate, you have the sugar, phosphate, and a nitrogenous base. And then you have that base, and the base has, you know, an A or a T. So let's say it's supposed to have an A, but a C got put in, maybe a C. That's a base substitution. So that's the most common thing that can happen. Only one base pair is changed. That's called point mutation. So if you're just getting rid of one, that's point mutation. There are 3 possible base mutations. And the teacher did, when we did our homework, that was part of the, you know, that was part of the answer, you know, the 3 possible base mutations. So let's go over them. One is silent mutation. A base substitution has occurred. The resulting codon is still coding for the same amino acid as the wild type. So remember, the wild is what happens normally in the wild. So a silent mutation is, although there was a substitution, it resulted in the same protein, okay? So it's the same amino acid, same protein. So it wasn't noticed. Silent mutation. Note, redundancy. More than one codon can code for the same amino acid. I don't think I have the sheet. I think it was in chapter 7. Let me see. There was a chart. Here it is. So what they mean by redundancy, let me find, for instance, valine. So if you notice valine, so there are different codons that code for the same amino acid. So that's called redundancy. More than one codon can code for the same amino acid. Okay? Then you have something called a missense mutation, and that results when the altered codon codes for a different amino acid. So you know, an A got changed to a G, and what happens is it is coding for a completely different amino acid. It's going to make a different protein. So this can make the cell grow slow because the protein only functions partially. You know, it's not quite moving right, and it can result in what's called a leaky mutation. Okay, next is a nonsense mutation. Nonsense. Occurring when the base substitutions create a stop codon, resulting in a shorter, often non-functioning protein. Okay, so the 3 types are silent mutation. Although there was a base substitution, it's silent. It codes in the same amino acid or protein as the wild type. Looks just the same. Missense mutation, where it's obvious, it's like messed up. Those A's and T's or just whatever, it's making everything messed up, and the protein's only going to function partially. And nonsense, and you can think there's no nonsense, so it's stopping. You're using that stop codon. Okay. Null or knockout mutation, it totally inactivates the gene. They didn't really talk about that beyond that. I mean, null is like null and void, so it's done. Knockout, bam, knockout. So it totally inactivates the gene. All right, here's a question time. What bacterial environment does base substitutions happen most in? What bacterial environment does base substitutions happen in? So if you have a bacterial environment and that base substitution, so you substituted like an A for a T, when does this happen? It happens in aerobic aerobic environments. The air. It happens in the air environment. Aerobic environments. But why? And it's because aerobic O2 bacteria produce ROS. Do you remember ROS was that toxic, I don't remember what it stands for, but it was toxic. It was like bad for the cell. And so then enzymes that convert ROS to H2O2, which is hydrogen peroxide and O2, these chemicals oxidize the G, the guanine. So all these chemicals oxidize the guanine because it's an aerobic environment with that aerobic bacteria. Damn. A base pair, so guanine, a base pair. As a result, DNA polymerase, so remember DNA polymerase kind of comes along, so DNA polymerase tries to fix the problem, but makes a mistake. You have an oxidized guanine and DNA polymerase matches and makes an A rather than a C. Whoops, DNA polymerase, you fucked up. So let's pick that apart again. So we have this aerobic environment, and there was a base substitution that got messed up because ROS, and then the enzyme converted that to hydrogen peroxide and O2. These chemicals oxidize guanine, a base pair, right? So it oxidizes guanine. As a result, DNA polymerase tries to fix the problem, but makes a mistake. So a G got put in, and DNA polymerase is DNA polymerase tries to fix the problem, but makes a mistake. You have an oxidized guanine, and DNA polymerase matches and makes okay. So I wish I had a piece of paper. Okay. So what that means is you have an oxidized G, and so DNA polymerase comes along and makes an A, but it should be a C, right? Because it's A T, and then that should be C G, right? So DNA polymerase didn't really recognize the G because it was oxidized. It got all oxidized and was like, oh no, do I make an A? So DNA polymerase fucked that up, right? So I wonder if I'm allowed to curse on YouTube. I guess I should be careful. DNA polymerase messed that up. So DNA polymerase, you have an oxidized guanine, and DNA polymerase matches and makes a mistake. So it makes it like an A rather than a C. And so that's what happens, you know. So the G was already there, but it was oxidized because of those the hydrogen peroxide that was from the ROS that was a result from the, you know, the air. So if you work in order, you have air, you have bacteria, you have the toxic ROS that got converted to hydrogen peroxide and O2 that then oxidized the guanine that so when DNA polymerase was trying to complement that guanine, it didn't really recognize it because it was oxidized, so it gave it an A instead of a C. It needs a C. DNA polymerase messed that up. All right, so what are we gonna do? Moving on. Deletion or addition of nucleotides. So essentially, we messed up. What's going to happen? Deletion or addition of nucleotides during DNA replication results in mutation. I really should kind of separate that. So essentially, during DNA replication, it can result in a mutation. You can add or delete 3 nucleotide pairs, like AAG, or one codon, like just A, depending on what it was can be a big deal or a small deal. So depending on was it an A, was it a T, was it a C, was it a G, depending on what it was, where it is, it could be a big deal or a small deal. When you add or delete nucleotide pairs, it causes what's called frame shift mutation. This changes the reading frame of mRNA. Frame shift can result in a shortened or non-functional protein called knockout mutation, and it can result in premature stop codon. So the teacher actually went over this, and her example was like much better. I don't remember how many different things that she had, but essentially, you have all of this right here, right? A's, and so this should be a T, for instance, here, and this should be a T right here. But so if there was a deletion, so instead of the frame being this, there was a deletion, and so now the frame is, I don't know, this. So that could code for a completely different amino acid. And so you could also add something. So you could add a C, for instance. So that's what frame shift is. And the teacher kind of showed all the different ways, but it would take me a minute to write it out. So I'm not going to worry about it. You guys